Welcome to the deep dive. Today, we're plunging into a world where, well, science fiction is rapidly becoming surgical reality.
They are.
We're tackling what might be the greatest logistical, maybe even medical, failure of modern medicine, the huge shortage of organs for transplantation.
Yeah, it's a massive global health issue.
Yeah.
I mean, the lack of tissues and organs, it's a huge driver of premature death. It's even estimated to be the leading preventable cause of death in America.
Preventable,
And
wow.
if you're a patient waiting, you're not just waiting days. Often it's years. Think about the UK. The average wait time for a kidney is 944 days.
Almost 3 years? Just waiting. That's devastating.
It is.
So our mission today in this deep dive is really to explore this, well, audacious technology that promises to maybe erase those waiting lists entirely.
Yeah.
3D bioprinting and advanced tissue engineering.
Right. We're looking at a fundamental shift, moving from relying purely on donations
Right.
to actually creating what's needed.
Mhm.
Exactly, creation. And at its core, 3D bioprinting isn't magic. It's an additive manufacturing process. Think 3D printing, but instead of plastic.
You're using living cells.
Living cell suspensions, yeah, combined with supportive biomaterials, building complex three-dimensional structures, layer by tiny layer.
Okay, so the idea of printing organs feels incredibly futuristic, but you're saying the basic printing part isn't new.
Not at all. The technical concept, that goes back to 1986. Charles W. Hull's stereolithography.
Stereolithography.
Basically using UV light to solidify thin layers one after another to build a 3D object. What made it bioprinting was the rise of computer-aided design, CAD software.
Ah, the design aspect.
Precisely. That lets researchers create this perfect digital blueprint of an organ, customized right down to the micro level for a specific patient.
Got it. Okay, so let's unpack the core building block then, the bioink. It sounds simple, like something for my desk printer, but I guess it's way more complex.
Oh, infinitely more complex. It's absolutely key to the whole thing. Bioinks are, well, they're intricate mixtures. You've got living cells, obviously,
Right.
then important biomolecules, and, crucially, supportive scaffolding materials, usually hydrogels.
Hydrogels, like jelly.
Sort of, yeah. They can be natural polymers, like alginate from seaweed, or synthetic ones like polyethylene glycol. They have to give immediate structure, hold things together while the cells start building their own matrix.
The requirements for this ink must be incredibly tricky, though. It has to be, what, liquid enough to squeeze through a tiny nozzle?
Exactly.
But then instantly firm enough not to just collapse into a puddle.
You've hit the engineering paradox right there.
Yeah.
And it has to mimic the body,
Yeah.
which means being highly hydrated. Our bodies are about 60% water, right? So hydrogels are perfect because they hold massive amounts of water. Then, for solidification, they often use photochemical reactions. They print the layer, flash it with a specific light, often blue light.
Why blue light?
It tends to be less damaging to the cells. That light triggers the gel to cross-link, basically. It becomes elastic and solid, trapping the cells safely inside without cooking them.
It's amazing the materials they're experimenting with. I read one source talking about using something like spider silk protein.
Yeah, that's a fantastic example of biomimicry, looking to nature. Spider silk is being mixed with cells, like mouse fibroblast cells in some studies.
Because it's strong.
Incredibly strong and resilient. It offers amazing structural and mechanical properties, and early results suggest it could be really promising for regenerating new functional heart muscle tissue.
Wow. Okay. And the cells themselves, where do they get those? Is it like a biopsy from the patient?
That's the ideal scenario, yes. Using autologous cells the patient's own. That massively reduces the risk of immune rejection.
Makes sense.
The alternatives involve stem cells, either induced pluripotent stem cells, iPSCs. Those are adult cells reprogrammed back to an embryonic-like state. Or human embryonic stem cells, HESCs. But, and this is crucial, using HESCs immediately runs into significant ethical debates because the research often involves the destruction of human embryos.
Right. A whole other layer of complexity there. Okay. Let's shift from the ingredients to the actual results. Printing a whole complex organ like a heart is clearly the ultimate goal. But what about simpler tissues? Are any of those getting close to actual clinical use?
Yeah, researchers are, I'd say, cautiously optimistic. Simpler structures, tissues that don't need that deep network of blood vessels or high metabolic rates, they might be maybe 5 to 10 years away from wider use. That's what some people in the field think.
Okay, 5 to 10 years. What kinds of tissues are we talking about?
Cartilage and bone seem to be leading the charge. We've seen successful creation of cartilage scaffolds loaded with cartilage cells, chondrocytes.
Like for knee repairs.
Potentially, yes.
Yeah.
There was even a key study where they 3D printed human cartilage and successfully implanted it into mice, and it integrated. And for bone, there's interesting work like the nano-kicking method from the University of Glasgow. They essentially vibrate stem cells in a way that encourages them to turn into bone cells and build bone structures.
Nano kicking sounds energetic. And what about skin? Structurally, it's layered, but maybe less complex internally than a kidney?
Skin is relatively more straightforward in some ways,
Hmm.
yeah. Bioprinting is great for it because you can precisely lay down those different layers, the keratinocytes on top, fibroblasts underneath, melanocytes for pigment.
So for burn victims.
Exactly. It could be revolutionary for treating really extensive, deep third-degree burns where you just don't have enough donor skin.
That's incredible progress on simpler structures, but now we have to hit the big wall, right? The heart, lungs, liver, kidneys. What is the single biggest technical hurdle stopping us from printing those?
It's one word, really. Vascularization.
Blood vessels.
Blood vessels. Everywhere. For any complex tissue, anything metabolically active, to survive longer than a few hours or days, every single cell needs to be super close to a blood supply. We're talking within 100 to 150 microns,
How small is that?
less than the width of a human hair. Imagine printing a branching network of capillaries, tubes often smaller than the cells themselves, throughout a whole organ. It's been called nearly impossible.
Nearly impossible.
It's such a barrier that NASA even put up a big prize, the Vascular Tissue Challenge, $500,000, just for keeping a thick piece of engineered tissue alive, proving it had functional blood vessels.
Okay, if it's that hard, how are the cutting-edge labs even trying to tackle it? You can't just print tiny pipes, can you?
You really can't just print simple pipes. It has to mimic nature's complexity.
Whoa.
Harvard developed a pretty amazing method called co-SWIFT.
Co-Swiss.
Stands for coaxial sacrificial writing in functional tissue. It basically prints hollow channels that become blood vessels, and it can do it with a multi-layer structure, the smooth muscle cells, the endothelial lining cells.
So like a real blood vessel.
Getting there, yes. And they've managed to embed this vascular network into spheres of human heart tissue that actually beat synchronously.
Wow. Okay, but designing that intricate network for a whole heart, that must have millions of vessels. How long does just the design take?
That's where computational power comes in.
Hmm.
Stanford researchers developed an algorithm specifically for this. It can map out a vascular tree model for a human heart with about 1 million vessels.
Okay, and the time.
Get this. It's 200 times faster than previous methods. It cut down a task that could take months into about 5 hours.
5 hours. That's insane. So it's this combination of micro-level engineering and really powerful software.
Exactly. That seems to be the path forward for complexity.
Right.
And we're seeing progress elsewhere too, like in airways.
Airways, like windpipe.
Yeah, tracheographs specifically. There were studies in rabbits using dual-head printers, alternating different biopolymers to build the structure. The results were much better than earlier attempts. No complications, no narrowing or stenosis after they implanted them.
Okay, so 3D printing is making strides, but you mentioned there are other, maybe parallel approaches happening too, using existing structures.
Yes, exactly. While 3D printing aims to build from scratch, there are two other key strategies that sort of leverage what nature has already built, kind of biological shortcuts.
Shortcuts, I like the sound of that. What's the first one?
It's called decellularization. Imagine you take an organ from a cadaver, say a heart or a lung.
Okay.
Then you use special detergents to wash away all the donor cells. Everything, DNA, proteins, all of it. What you're left with is just the natural scaffold, the extracellular matrix, or ECM.
So you keep the building's framework, but get rid of the old occupants.
That's a perfect analogy. You keep the perfect native architecture, including the entire network of blood vessels, which is the hardest part to build.
And then?
Then you take that clean scaffold and recellularize it. You seed it with patients' own stem cells. They move in, multiply, differentiate. And the goal is you end up with a personalized functional organ using the original blueprint.
With basically no risk of rejection, because it's the patient's own cells.
Exactly. It bypasses the need to print that incredibly complex scaffold.
That's brilliant. Okay, what's the other parallel approach?
That one's xenotransplantation,
Yeah.
which basically means using components from other species.
Like the pig heart transplants we heard about.
That's one aspect, yes. But there's also fascinating work using animals in a different way. There was a study where they took human lungs that had been rejected for transplant organs that would normally be discarded,
Okay.
and they connected these lungs to the circulatory system of a living, anesthetized pig for 24 hours.
hooked human lungs up to a pig. Why?
The pig's blood flow and physiological support actually revived the lungs. It improved their function, their ability to exchange oxygen. The idea is maybe this could rescue organs we currently have to throw away, potentially tripling the supply of usable donor lungs.
Wow, okay. Using pigs as sort of temporary life support for organs. Amazing. So decellularization and xenotransplantation are happening now. What's the really futuristic concept? The next big leap?
That would probably be 4D bioprinting.
4D. What's the fourth dimension?
Time. With 4D bioprinting, you don't just print a static 3D structure, you print a structure that's programmed to change its shape or its function after it's printed, usually in response to a specific stimulus.
Like what?
Could be UV light, heat, maybe changes in pH or osmotic pressure, so the structure might fold itself or expand or become mechanically stronger after it's been implanted. It adds another layer of control and potential functionality.
Okay, my mind is slightly blown. But let's pull back from transplantation for a second. Are there other uses for 3D bioprinting that are actually happening now? Maybe less flashy, but still important?
Oh, absolutely. The non-clinical applications are huge and maybe even more immediately impactful in some ways. The big ones are disease modeling and drug screening.
Disease modeling, like printing tiny tumors.
Exactly. Scientists are mass-producing highly accurate 3D models of complex diseases. Things like triple-negative breast cancer models, cervical cancer models. They can then use these for testing new drugs, understanding the disease better, developing cell therapies.
And why is a 3D model better for drug testing than, say, cells in a petri dish?
Because cells in a flat 2D petri dish just don't behave like they do in the complex environment of the human body. These 3D mini-tissues, sometimes called organ-on-a-chip systems, they much better mimic that in vivo microenvironment.
So the drug test results are more reliable?
Much more reliable, yes. It drastically improves the accuracy of predicting whether a drug will actually work in humans. Plus, it helps reduce the need for animal testing and toxicology screening, which also addresses some big ethical concerns.
Right, okay. So the science is incredible, the potential is obvious. But moving this from a lab to a hospital, that's never just about the science, is it? What are the big non-technical roadblocks? I'm guessing cost is a major one.
Oh, it's staggering. Let's talk US dollars just for clarity. A sophisticated bioprinter itself can easily cost over $185,000.
Okay, pricey, but maybe manageable for a big institution.
Maybe. But the real killer is the consumables, the bioinks. Depending on what you need, how specialized it is, a single gram of bioink could range from maybe $4 all the way up to $100,000.
$100,000 for a gram of ink.
Yep, for highly specialized functionalized inks.
Okay, that explains why venture capitalists might be hesitant, especially with the long development times.
Exactly. VCs look at that timeline, often 10 years plus, just to get near human trials. And then they see the incredibly complex FDA regulatory hurdles here in the US, and many just balk.
Is there an example of that?
Yeah, Tivito Biodevices, a startup focused on printing nipple grafts and potentially breast tissue for reconstruction after mastectomy. They estimated needing something like $40 million and another 7 years of testing just to get their first product into human trials.
40 million in 7 years. So how do companies like that even survive? If the big goal, like printing a liver, is decades away, where does the money come from now?
They often have to pivot, at least initially. To fund the long-term, really ambitious research, many companies start by focusing on simpler products, things with a clearer, maybe faster regulatory path and quicker potential for profit.
Like what?
Often things in the aesthetic, or you might say vanity space, like the nipple grafts Tivito was working on, or maybe tissues for cosmetic breast augmentation. The idea is to generate revenue from these simpler applications.
To pay for the moonshot research on complex organs.
Pretty much, it's a pragmatic, if maybe slightly jarring reality of funding this kind of deep tech.
That is a harsh reality check, and it leads us straight into the ethical minefield, doesn't it? The tech is clearly moving faster than the rules and regulations. Let's start with safety. How on earth do you run a standard clinical trial on an organ that's custom-made for one person?
You really can't, not in the traditional sense. That's a fundamental issue. A bioprinted organ is the ultimate personalized medicine. It's bespoke.
So you can't test it on person B before giving it to person A.
Exactly, because it's made from person A's cells, tailored to their anatomy. This makes the standard pre-market safety testing, where you test on many people first, essentially impossible. How do you ensure safety before the very first implant? It's a huge challenge.
And if or when this tech becomes available, that cost barrier we talked about, that raises massive equity questions, surely. Who actually gets access?
Yeah, the sources we looked at are very clear on this risk. The incredibly high cost could easily deepen existing social inequalities in healthcare. The major concern is that access might be limited, at least initially, primarily to the wealthiest groups.
Creating another tier of healthcare access?
Potentially, yes,
Yeah.
exacerbating the disparities we already see in transplantation and beyond.
Okay, now let's talk ownership. This is mind-bending. If I provide my cells, but a company provides the fancy bioink, the complex software, the printer, who actually owns the finished organ?
That is a massive legal and ethical gray area right now. Who owns the intellectual property? Who owns the physical product? We have some legal precedents, like the famous Moore v. Regents case.
What did that say?
Broadly, it suggested that once your cells are removed from your body, you generally don't retain ownership rights over them or what's developed from them.
But wait, if you apply that to a life-saving organ built from my cells, you're saying I might not legally own the organ keeping me alive. That seems fundamentally wrong. Doesn't that clash with patient autonomy?
It absolutely clashes. The sources really highlight that tension. Who holds the rights? Who holds the liability if something goes wrong? Is it the patient's biology failing or the company's manufactured product? It's legally uncharted territory.
A legal minefield indeed. And one last thing, informed consent. If the science is this complicated and the risks are so unknown because it's custom, how can a doctor possibly explain it all properly for a patient to give truly informed consent?
It's incredibly difficult. Just the sheer complexity of the technology makes it hard to explain comprehensively. But more than that, because the field is so new and each product is unique, the long-term risks for that specific patient's custom organ are largely unknown.
So you can't fully list the potential downsides.
Not with the same certainty as established treatments. This really compromises the ideal of fully informed consent, where the patient understands all the potential risks and benefits before agreeing.
So wrapping this up, this deep dive has shown this amazing duality. On one hand, this incredible life-saving promise of custom organs, no rejection, maybe ending waiting lists.
The potential is immense.
But on the other hand, these huge walls, the scientific mountain of vascularization, the brutal financial realities, and this deep tangle of ethical and legal questions about safety, fairness, ownership.
It's a profound juxtaposition. And it forces us to think about the regulations, or lack thereof. These products, they don't fit neatly into existing boxes. They're part living biology from the patient, part synthetic materials like hydrogels, part complex software and hardware.
It's not just a drug, not just a device, not just a tissue transplant.
Exactly. It's something new. And that leaves you wondering, doesn't it? How should regulators, like the FDA or EMA, classify a personalized organ built from your cells, sophisticated materials, and AI-driven design? Is it a medical device, a biologic drug, a tissue product, or a whole new category?
And why does that classification matter so much?
Because that classification determines everything. The path for approval, the standards for safety and efficacy, who is liable if something goes wrong, and ultimately, how accessible this potentially revolutionary technology will be. It's the quiet, regulatory question that could shape the entire future of transplantation.